Hello, my name is Taylor Ryle, and for my MLS management project, I did an analysis on automated semen analyzer options for medical laboratory use for Covenant Healthcare's Houghton Laboratory in Saginaw, where I completed my clinical internship. Semen analysis is used to aid in the classification of a male's fertility status. This is done by looking at a variety of physical and chemical characteristics of semen specimens, including the specimen's volume, pH, and viscosity, as well as microscopically assessing the spermatozoa concentration, motility, and morphology. Semen analysis is in high demand, but unfortunately is also considered a high complexity test, meaning it takes a lot of expertise and time to complete. Although manual methods have been shown to produce reliable and quality results, an automated method may also be able to produce more consistent results as it eliminates variability between technologists' assessment. Covenant currently offers semen fertility analysis and semen post-vasectomy analysis, for which a manual method is used for both. In the last year, there were 368 semen analyses performed at Covenant. 288 of these were post-vasectomy analyses and 80 were fertility analyses. Specifically for semen fertility analysis, the volume, viscosity, and pH of the specimen is recorded, as well as microscopically looking at the sample to obtain a spermatozoa count, motility, and morphology. Although it should be noted that for the morphology assessment, only certain technologists have had special training to perform this, so it can take a few days to complete this test. For semen post-vasectomies, we are simply looking for the presence of spermatozoa, and if it is present, we record a count in the motility. The first automated system that I looked into is the SQA Vision by Medical Electronic Systems. The SQA Vision is an automated semen analyzer that can be used for fresh, IUI, washed, post-vasectomy, frozen, mail-in, longevity, and manual count semen sample types. It takes about 75 seconds for normal quality specimens to be analyzed and about 5 minutes for post-vasectomy specimens. The SQA Vision measures concentration, motility, and morphology of sperm automatically. Now on the right is a picture of the system and an example of a screen that could be viewed while doing an analysis. The SQA Vision measures the concentration of spermatozoa by relying on the fact that a very specific wavelength of light is absorbed by sperm in the concentration chamber of the analyzer. An optical density detector measures the amount of light absorbed by the sperm cells and then converts it to optical density, which is then converted to a concentration via a microprocessor. The motility is assessed on the SQA Vision because the sperm cells move through the light beam in the vision. The sperm cause a light disturbance, which are then converted to electronic signals, which are then converted to motility parameters via a microprocessor. In the pictures above, we can see an overview of this technology on the left, and on the right is an example of a graph that would be used to obtain a motility measurement. There are some consumables that come along with the SQA Vision. Not all of them are required, but they include a Quick Check DNA Fragmentation Index Kit, Quick Check QC beads, which come in 3 levels, a Quick Check Dilution Kit, which can be used to increase sample volume, so this would be helpful for short samples, Quick Check Liquefaction Kit, which promotes liquefaction of especially viscous samples, making it easier to analyze, Vision Fixed Cover Slip Slides, which disperses sample or QC specimen to one field of vision, Quick Check pH/white blood cell test strips, which are the only pH/white blood cell strip that are specifically designed for semen that are also approved by the FDA. And there's also QC that goes along with those. Quick Check one-step vitality stain, Quick Check pre-stained sperm morphology slides, which have a thin coating of stain on them so the sperm absorb them on contact, which is how the sperm morphology is assessed. Quick Check Beads Proficiency and Training Kit, which goes along with the initial validation process, which I will discuss, and the Quick Check Beads Precision and Linearity Kit, which is used in the semi-annual mini validation, which I will also discuss. And on the right are just some pictures of these products. Maintenance and QC. According to MES, there is a daily maintenance. It just includes cleaning the measurement compartment daily and after every 10 to 15 samples run with the manufacturer's cleaning kit. For quality control, there is an internal quality control, which is an electronic self-test and auto-calibration. This runs automatically after startup, and the reference values are also verified prior to each test. For an external quality control, they recommend the use of quick check beads for concentration and negative control, and then for concentration motility measurements, the use of latex beads or a stabilized sperm CAP. So the overview of the validation process, there was a lot of information on the MES website about the SQA Vision's process for validation. I was not as successful in finding this for the other systems, so I would just like to note that although we don't know specifically what that would entail for the other ones, we can assume it might be something similar to this. For the initial validation process, it would consist of running 20 patient samples of different concentrations, both manually and on the analyzer, then comparing the results. MES recommends that you send them the first 5 samples for an initial overview before completing the rest to see if they recommend any modification of techniques or troubleshooting if needed to make sure that the measurements are comparable. For the replication and detection limit experiment for precision, they recommend 10 runs of positive QC material on 2 levels and 10 negative QC runs to accurately assess the random error associated with the method. 2 live patient samples should also be run on the patient mode to assess morphology and motility. For linearity and reportable range experiments, it would consist of preparing 6 samples compiled from multiple specimens to achieve 6 levels of concentrations using the quick check dilution, and then running those to get some results to look at. They also had some information on the validation for post-vasectomy analysis. So according to MES, this would include recording the pH and white blood cells using the quick check pH strips, and then testing 20 post-vasectomy samples without centrifuging first, and then if no sperm is found, centrifuging and viewing them again, and then doing this simultaneously on the analyzer and under a microscope viewing 50 fields to then compare the data from both to see if they get comparable results. There's also some information on a semi-annual instrument calibration confirmation. MES recommends that twice a year the calibration be tested against the factor calibration parameters as a proactive maintenance measure. There's also a semi-annual CAP challenge. The CAP will have a specific sample set that is peer-reviewed and compared to other users. There wasn't a lot of specific information available on this. The website said to contact CAP for more information. There's also a semi-annual system mini validation / motility and morphology confirmation. So it's highly recommended that the facility confirms the precision and lower limit of detection by completing an abbreviated validation study semi-annually. And again, not a ton of information on this. They said to contact MES directly for more information, but they also recommended that 5 samples are tested manually and on the analyzer and compared either annually or semi-annually. I also found some literature that discussed the SQA Vision. So there was a double-blind study that found that there were no discernible differences between the SQA Vision analysis and manual analysis of total sperm motility and concentration, but there was a disagreement between the measurement of progressive motility between the 2 methods. The sensitivity and specificity for both the manual and automated systems were both within acceptable limits, but there was a difference between the morphology assessment between the manual and automated methods. And on the right is just some graphs from one of the articles that I read about the SQA that just shows where all these parameters line up. The next automated system that I looked at was the SCA scope by Microptic. It is called the first totally automated device for human semen assessment. It measures motility and concentration, morphology, volume, pH, leukocytes, and more using artificial intelligence with the assistance of phase microscopy. This system can also run multiple patients at once, and it also has an option for manual counting if needed. On the right is just a picture of the system, so you would load it into the machine there, and then it would show you the results on a desktop. There are also some consumables that go along with the SCA scope. There are counting chambers, which are used for the motility, concentration, and pH measurement, QC beads, morphology slides, which are pre-stained, mounting medium for slides, which helps to obtain a contrasted image after staining and also helps slides dry faster, gold cytoDNA, which is a DNA fragmentation kit, vitality slides, and leukocyte slides, which are a kit for detection of peroxidase positive cells in semen. On the right are just some pictures of those items. I also found some literature discussing the SCA scope. So there have been conflicting conclusions found on the validity of the SCA scope. There were some studies that showed an overestimation of sperm count and other studies that found no significant difference. Studies have also shown that the SCA scope has a higher repeatability of counts in cases of azoospermia and oligospermia, so cases where there are no sperm present normally or a low count, and also a high repeatability for progressive motility measurements. Like most CASA systems, which means a computer-assisted semen analysis system, there was a significant disagreement between the SCA analysis and manual analysis of morphology. The last system that I looked at was the LensHook X1 Pro by Hamilton Thorne. This is semi-automated and is called the world's smallest semen quality CASA. It uses optical design and image analysis combined with artificial intelligence to assess pH, concentration, morphology, and motility. Sperm concentration is measured using sperm unit density. Motility is assessed by tracing sperm trajectories. Sperm morphology is assessed using head and tail percentage, and pH is measured by analyzing an image for its saturation and brightness. And it connects to a pre-existing desktop for visualization of specimens. So as you can see on the right, you would load the specimen in, and then it does give you some information on top, but you could actually view the specimen on a desktop. The consumables that are included with the LensHook X1 Pro, there's an internal quality control. This comes in 3 levels. There's beads in a slide, and then there's also a seam test slide and a QC video, which helps to look at the precision of the video and accuracy. There's also sperm separation solution, semen test cassettes, liquefaction test cup, and sperm DNA fragmentation tests. So then I look at some literature discussing the LensHook X1 Pro. So there was a validation study performed by Cleveland Clinic that found that semen analysis using the LensHook X1 Pro took 90% less time than a manual semen analysis. The same study found that the analyzer had a high specificity and sensitivity for parameters such as motility, progressive motility, and concentration compared to manual methods, but the analyzer makes it a simpler process and takes a lot less time, which makes it more efficient and cost-friendly. And then this is just another diagram from Hamilton Thorne showing how the manual method and the LensHook method compare to each other. I could not find any specific pricing information on any of these analyzers. They all said to contact the manufacturer directly for more information on that, which I chose not to do because I assumed they would be asking me things that I did not have access to, and I also did not want them calling the lab unless the lab was truly interested in their product. The closest thing I could find was Fisher-Price did have the SQA Vision listed on their website for $32,310, but again, this might not be a good reflection of the true cost. It can be assumed that using an automated analyzer for semen analysis could cut down on the cost of labor for a technologist having to take the time to manually analyze the specimen. And it would also maybe save some money on the supplies needed, depending on which system we went for. So some final remarks as I conclude this presentation. Although manual and automated semen analysis are both reliable options for the assessment of pH, concentration, motility, and morphology and volume of semen samples, Covenant might find it advantageous to invest in an automated semen analysis system, as they both save time in the lab and reduce turnaround time. They also produce more consistent results than manual analysis. There are many automated semen analysis options on the market, including the ones that I looked at, the SQA Vision by Medical Electronic Systems, the SEA Scope by Microptic, and the Lenshook Pro X1 by Hamilton Thorne. And they've all been shown to produce comparable results to the manual method, and there's probably some other great system options out there. Both semen fertility testing and semen post-vasectomy testing are in high demand and are crucial for the assessment of men's reproductive health. So it is important that the efficiency and accuracy of the results that we produce are at the forefront because they are vital. And then these are just my references.